I am Mark Newman. I'm going to introduce you to some concepts and some design features that we're using at GeoVax to address the evolution of SARS-CoV-2 variants, and specifically to induce broader immune response that might provide a more universal coronavirus coverage. Now, part of this, I'm going to first of all introduce you or remind you about immune responses to viral infections, and with a specific focus on SARS-CoV-2. Introduced you to the concept of targeting conserved viral regions as opposed to more variable gene products. Then I'll introduce you to the GeoVax MVA-VLP technology that we're using to design and produce vaccines. And I'll show you some data on our first generation vaccines targeting COVID. And then I'm going to close with some design features that we think might be applicable to the production of vaccines that will provide true universal coronavirus coverage. Now, immune responses to viruses are complex, and successful immune control of viral infections include antibodies, which are specific to virus glycoproteins typically. They neutralize the virus or block the virus from infection, and they also limit the cell-to-cell spread. Cellular immune responses are mediated by T lymphocytes, both cytotoxic or CD8s and helper T lymphocytes, CD4. T cells directly act to limit viral replication, generally through the function of cytokines, interferons, things like that, but they'll also mediate clearance of infection by killing virus-infected cells. A truly effective immune response that one would see after recovering from a viral infection will be multifunctional with both antibody and T cell responses. And this is really required for optimum protection from infection and for limitation of potential serious illness. This is the goal of a vaccine, to induce multifunctional responses. Now, SARS-CoV-2 antibody responses have been well characterized over the last year. First of all, neutralizing antibodies specific to the spike protein are present in the sera of most convalescent patients. Neutralizing antibody will persist for months to maybe even years. Long-term data is still absent just because the pandemic has only been ongoing for approximately 2 years. SARS-CoV-2 infection does induce memory B lymphocytes, which are resident in the plasma cells. So the indication that some level immunological memory in the B cell compartment is induced. Right now, we know that memory B cells can be readily detected for 8, 10, 12 months. Long-term studies are still underway. Now, the duration of neutralizing antibodies might be short-lived. There are several reports indicating this. And certainly, if you look at seasonal coronaviruses, long-term neutralizing antibodies are really not a feature of recovering from these infections. There has been some concern over non-protective antibodies that would actually augment disease, antibody-mediated disease enhancement. However, this has not really been observed much, so it's not considered a significant fear with most of the vaccine strategies being employed. Now, the highly immunogenic nature of the S protein, the spike protein, plus the fact that it has the receptor binding domain means it is the optimum antigenic target for inducing antibody responses that will neutralize the virus. And of course, this is what's been used in the first generation vaccines by all of the major vaccine producers. Here, the focus is primarily on the induction of neutralizing antibodies. Now, there's a concern with neutralizing antibodies in that SARS, as all RNA viruses, is capable of rapidly mutating and literally can evolve and generate variants, which may either be more transmissible or resistant to antibody neutralization if these variants occur within the S protein, the spike protein. This little graph here, this figure here, just simply shows the level of genetic variation that we're seeing. This is early in the pandemic, from December 2019 to February 2020, which you can see is a lot of variation is showing up with the SARS-CoV-2 as it spreads through the population. And of course, if these mutations are in the S protein, then there's a chance that it'll impact the efficacy of neutralizing antibodies. This, in fact, has been shown to happen. Here I'm showing you the neutralizing titers that were induced by both the Moderna and Pfizer vaccine against an early variant, the alpha variant, B.1.1.7, which is hold up pretty well against the Washington variant. But then by the time we get to the beta variant, which is the B.1.351 South African variant, you can see that we're seeing a diminution in the effectiveness of the neutralizing antibody responses. So this is a concern. The variations will accumulate within the virus population as it spreads and limit the effectiveness of neutralizing antibody responses that are induced by the vaccines. Now, the number of variants continues to grow. Just make a couple of points here. These are the variants of World Health Organization concern. And I'm showing, just top 8. Of course, the Delta variant is the one that most people are being infected with currently. But we started with the Wuhan strain, then the UK variant, also known as Alpha, the Beta variant, South African, and all of these are accumulating mutations within the S protein, 3, 8, 10, 7 different mutations. These mutations appear to be increasing the infectivity and potentially transmissibility of the viruses, and they are increasing the resistance of these new variants to the neutralizing activities of antibodies induced by both vaccination and some of the human monoclonal antibodies. So these variants are happening and they are presenting a concern. As noted here, the Delta variant is the most problematic at this stage of the game, and it's only number 4. There's many more already in line. Now, how do you address variants of world health concern? Through vaccines. Well, one of the approaches is to have a global surveillance system, similar to what is used for influenza. So here you can track the emergence of different variants and then adjust your vaccine using a protein, the most common S protein sequences that are circulating at the time. So this might require yearly or regionally campaigns to adjust the virus vaccine, and theoretically would involve yearly booster immunization campaigns. So again, similar to something that we see for influenza. But the COVID vaccine or COVID-2 seems to be mutating even more quickly than influenza. So it's a significant concern. An alternative approach is develop vaccines that are specific to viral proteins that are less subject to variation. So these can be ancestral sequences or matrix sequences if you want to target a protein like the S, therefore induce antibody responses that would recognize more broadly the types of circulating coronaviruses that are out there. Or, as I'm going to show you, you can target more conserved regions of the virus, specifically target T-cell epitopes, thereby broadening the immune responses so that the vaccines will induce T-cells and antibodies. Now, what do we know about T-cell immunity? So in the coronavirus system, or CoV-2, it's similar to antibodies in that T-cell responses are readily detectable in 90% or more of convalescent patients. So patients that have recovered mostly have CD4 responses. That's the predominant T-cell response, but we also see significant CD8 responses. Multi-specific T-cell responses, meaning they target multiple epitopes, and multifunctionality of T-cell responses, T-cells that are producing both interferons and cytokines such as IL-2 are associated with accelerated viral clearance and therefore with protection from severe disease. So this is the goal when generating a vaccine response. Now, interestingly, while we're concerned about the duration of antibody responses, patients who recover from SARS-CoV-1, which is an outbreak that happened in 2003, have been shown to have long-lasting memory T cells. So 17 years after the outbreak, memory T cells can still be detected in SARS-CoV-1 patients. Now, this graphic is interesting. That what it shows you is that individuals who have functional T cells early on after infection actually have a much less or a reduced severity of disease. So if T cells are readily detectable in days 2, 3, 4, and 5 after infection, disease is less likely to be severe. Alternatively, if immune responses would come up with later, as in day 10, 11, 12, what we're seeing are significantly higher levels or higher severity of disease. So if an early T cell response can actually help protect individuals, whereas an antibody response and a T cell response coming up at a similar time frame, later in infection, correlated with a higher disease severity. Now, where do these early responding T cells come from? Well, they could be from an asymptomatic CoV-2 infection, but they can also be from exposure to coronaviruses other than CoV-2. T cells in uninfected donors that recognize epitopes in different proteins, primarily non-structural proteins, have been readily identified. So these are individuals who have not been infected with CoV-2, and yet they have T cells that recognize SARS-CoV-2 epitopes. The suggestion is that these are probably T cells that were induced against the seasonal coronaviruses, and these epitopes are conserved within many coronaviruses, and therefore they serve as the first line of the immune response. Specifically, they're in non-structural proteins. So, for example, the non-structural protein 7 has multiple T cell epitopes that are highly conserved amongst all or most animal beta coronaviruses. So the hypothesis here is that vaccine induction of T cells specific to conserved epitopes is likely to be highly beneficial and potentially even required for optimal and long-term efficacy as SARS-CoV-2 variants continue to emerge. So how do you induce responses and what do you target? Well, what we would target for T cell responses would be highly conserved sequences. And if you look at the genome of multiple CoV-2-like viruses, multiple coronaviruses, what you can see is there's a lot of variation in the S protein, the spike protein, plus some of the other open reading frames. But most of the open reading frame genes in open reading frame 1A and 1B, these are non-structural proteins, in fact, are highly conserved. Also, you notice that the envelope, the E protein, and the M protein, and the nucleocapsid, the N protein, structural proteins, are highly conserved. So there are a number of proteins within the virus that are not prone to great variation, certainly significantly less variation than the S protein. Now, does this variation make a difference beyond escape of the immune responses? And we believe that it probably does. There are reports, for example, that mutations in the S protein and the open reading frame 8 are actually what accumulated over time. And this facilitated or allowed the spread of CoV-1 from bats into civets and then ultimately into humans. So these mutations in the highly variable regions of the coronavirus protein certainly appear to be linked to changing in pathogenesis and the types of species that can be infected with coronaviruses. Of interest, MERS-CoV coronavirus exhibits an extremely high sequence homology amongst the open reading frame 1A and 1B genes, but a great deal of mutations within the S gene, which, the MERS virus doesn't even use the same receptor as CoV-2. So this might be expected. So by targeting these conservative regions, it may be feasible to really target a broad range of beta coronaviruses. Here's some real data that shows how important this might be. Here we have T lymphocytes being tested, both CD4 and CD8 cells, from unexposed individuals, individuals who are not infected with SARS-CoV-2, and exposed and recovered individuals, in which you can see primarily here in the yellow part of these circle diagrams is these are T cell responses that recognize epitopes that are in the open reading frame 1A, 1B, the non-structural proteins, and which you can see in unexposed individuals that the majority of the epitopes that are recognized by these T lymphocytes that are also recognized by epitopes in SARS-CoV-2 are specific to these open reading frame, these non-structural proteins. However, there's also a significant amount of cross-reactivity to the S protein, as shown in the red part of the diagrams, and then also the membrane and to a lesser degree nucleocapsid. So there are conserved regions and conserved epitopes within S. There are conserved regions within the membrane, the M protein, but the bulk of these are apparently in the non-structural proteins, at least for inducing CD4 cells and CD8 T lymphocytes. So the potential of T cell responses to conserved epitopes is really quite high. Nucleocapsid, the N, membrane M, and envelope proteins are all highly conserved structural proteins. We know that these proteins have epitopes that are immunogenic in people, and in fact, T cells are detectable in convalescent SARS-CoV-2 patients. So there's a cross-reactive level of response. Those that are T cells probably induced by seasonal coronaviruses, but also they are induced as part of the recovery process with SARS-CoV-2. However, non-structural proteins are actually the most highly conserved. So non-structural and open reading frame proteins represent a rich source of T cell epitopes that we also, again, know are immunogenic and can induce cross-reactive T cell responses. So the hypothesis here when designing a vaccine is that targeting the N, M, and E proteins using vaccines are more likely to protect against emerging S variants as the pandemic continues. And if we can expand the T cell response so that we're also targeting open reading frame 1A and 1B non-structural proteins, we'll approach the level of universal coronavirus protection. So how do we do this? With GeoVax, we work with modified vaccinia virus ANCRA as a viral vector. We work with MVA because it has a large coding capacity. This allows us to encode multiple viral gene products in the same virus vector. Here, this represents what the MVA would look like, or it does look like, for our first generation CoV-2 vaccines. So antigen 1 would be the S protein. Antigen 2 is the membrane. Antigen 3 is the envelope. We specifically picked S, M, and E because these are the proteins that are required for assembly of the virus into a virus-like particle. So the MVA encoding these gene products will infect the cell within the host, and you'll actually get virus-like particles being produced, which are highly immunogenic and look authentically like the SARS-CoV-2 virus. Now, does this work? And in fact, yes, it does. See that the lead product here, referred to as CMO2, encodes a stabilized pre-fusion version of S, then membrane plus envelope, all in the same deletion site or cloning site of MVA, but under different promoters. Cells that are infected with this CMO2 MVA will produce VLP, as shown in the lower left-hand corner here. These are coronavirus-like, virus-like particles made up solely of S, M, and E without any of the other viral proteins. You look at the right side of the panel, we show that you can get spike detected by a Western blot in the media of cells that have been infected with the MVA CMO2 vaccine. The fact that they're present in the media indicates that it's either secreted or that we are getting virus-like particles formed. When the reason we're focusing on the virus-like particles is because of their increased immunogenicity. And then on the lower right hand, I'm showing an immunostaining of cells infected with the CMO2 vaccine, showing membrane expression. So CMO2 encoding membrane spike and envelope is expressing or producing virus-like particles that mimic the COVID-2, SARS-CoV-2, and induce immune responses. Now, are these immune responses effective and useful? I'm going to show you a couple of quick snapshots of some animal slides. First is an efficacy study in hamsters. And what you can see on the left panel is that one immunization or two immunizations induces high levels of IgG in immunized hamsters. On the right side, we're showing that these immune responses are in fact capable of mediating protection. This is a weight loss and recovery graphs shown of vaccinated and unvaccinated animals following an infection with SARS-CoV-2. So the types of responses that are induced do in fact protect animals from severe disease. Now, if we move to the human ACE2 transgenic mice, we're able to now look more carefully at neutralizing antibody responses and T cell responses. So on the left panel, what I'm showing here is that two immunizations with CMO2 induce significant levels of neutralizing antibody responses in most of the mice. On the right side, we're showing that we're seeing CD4 and CD8 T cell responses with either one or two immunizations of the CMO2 MVA vectored vaccine. Now, important I want to show here is that looking at the CD4 responses, you notice that we get significant CD4 responses to the S peptide pool after one, and it's boosted after two immunizations. These T cells also recognize a variant, the South African variant, or the alpha variant in this case of peptide pools. So we're seeing a significant amount of cross-reactivity even against the variant S protein. Now, the M peptides are also, M protein is also immunogenic, although at a lower level. We're seeing similar response characteristics for the CD8 responses, but here we're getting the maximum response with two immunizations. And we're seeing very significant levels of CD8 T cell responses against both the immunizing peptide and a variant, plus significant high-level CD8 responses to the membrane peptide. So we're able to target through cellular immune responses the conserved membrane protein, but also conserved regions within the S peptide. And on the bottom panel, I just want to emphasize these are effector memory and effector and central memory are primarily effector memory responses that we're measuring, but there is some central memory responses even in these short-term studies. Now, how does this translate into protection? And here, what I'm showing you is that we can induce 100% protection against lethal infection in the ACE2 transgenic mouse using either one dose, which are the line in the red, or using one dose is the line shown in the green, or two doses in the red. So the panel on the left shows the weight loss and recovery, and you can see the vaccinated animals just vacillate around the cutoff point. We do not have weight loss. 100% of the animals survive. And on the right side, this is actual percent survival. And again, a single immunization is inducing protection at the 100% level. Now, this is a homologous challenge model. So the vaccine was based on the Wuhan strain, and the challenge model was based on the Wuhan strain. So now the question is, well, what if we want to look at a variant? And here what we've done is challenge the animals with a South African variant, the B1351, which is in this model system significantly more virulent and somewhat more variable in terms of the S protein. And what you can see again on the left side is that animals, while they can become infected, only lose weight for a short period of time and rapidly rebound if they've been vaccinated. In this case, all animals were vaccinated twice. The right side shows the viability or survivability of the animals. So we're seeing over 80% animals surviving a lethal challenge with the South African strain, indicating a protection level that is outside of just the single variant, suggesting that T-cell responses to conserved regions are in fact providing a survival advantage. So how do we interpret the data? First of all, the use of MVA as a vector really supports the design and production of next generation vaccines, primarily because we can produce products that encode multiple viral proteins and therefore can induce a broader immune response. Here we're looking at the S protein as the antibody target and M and E proteins as the T cell targets, M and E being the more highly conserved proteins. Now, the combination of S, M, and E expression supports VLP formation, which should provide an optimal immunogenicity because the VLPs are highly immunogenic. They provide antigen in a concentrated form, and it's very much in a functional native form. Functional antibodies and T cell responses are induced to mediate protection based on the animal models that we've looked at. And this is with the endpoints of both lethality and pathogenesis. So the vaccine is very much capable of inducing both protection from disease and protection from death. Now, I showed you some data with protection against the South African variant. So how would we take it a step further? Here, what we would do is propose to start looking at some of the open reading frame, the non-structural proteins. These represent over 60% of the viral genome. So there's a lot of targets to work at. And as I showed you in the human CoV-2 studies, these encode a large number of T cell epitopes. So what way that we can build on these for the future is to use the existing CMO2 vaccine, which encodes the S, M, and E proteins, with S being the antibody target, M and E as being T cell targets, highly conserved T cell targets, but encode these non-structural or open reading frame proteins under different promoters. They don't have to be part of the VLP. So this is the next step as we move forward. And again, the goal is to induce T cell responses that are not impacted by the continual evolution of the more variable S protein. And with that, I want to close and just point out the team of GeoVax, Armand Domi producing the viruses, and Mary and her team doing immune responses. We work with Alex Pokrayev at University of Texas Medical Branch on the hamster studies, Mark Lewis at BioQual with Dr. Carr working on the transgenic mouse studies. Our work is funded in part by the NIH. And with that, I will close.